All right. Okay. Good morning, everyone. Um, I hope you're doing really well and you're having a good week. Um, just,
Estamos hablando.
all right. Gracias, Jesús Eduardo, to remind us that you have to keep your microphone off and also your camera is off. Second, I also remind people that we're going to leave questions to the end. And just a reminder to David, I will start my watch in around 11:48. I will probably ask you to stop, but I'm sure that you're gonna keep your own timing of your own slides. That's right. And without further ado, it is to me an enormous pleasure to introduce all of you David DeFilippis. David DeFilippis and I met when he was in undergrad, and my PhD professor was teaching something, and then he raised his hand and said, well, how do you do that? And Stefan explained how he was doing it, and he's like, I can do it in a better way. And we would just turn around and say, who is this undergrad that thinks he's so like cocky and can know all these things about ecology? And that's how his major saga with ecology began. But David has a really interesting life. He was a welder before, an industrial welder in the northern US and Canada. He's a mechanic. He's a diesel mechanic. He works with ginormous trucks. He also designed robots, and he taught himself how to program even before starting undergrad. And then we became really good friends. Afterwards, he joined academia and then decided not to do academia anymore. He left again to do software engineer, and then he came back to academia to do his PhD. And so it's always been great because his mind works in a different way. And me trained as a classic ecologist, then we sometimes kind of bump heads because sometimes ecologists think one way and programmers and mathematicians think another way. And I think those have clearly merged in David in a phenomenal way. So David works, he got his undergrad at University of Wisconsin-Milwaukee, and then he studied his PhD at Marquette University. And so we shared at the same lab for a few years. We became really good friends. And he has earned several very prestigious awards. One of them is the Graduate Assistance in Areas of National Need Fellowship, and other very prestigious NSF awards. And he works mainly with large forest data sets, and he uses different combinations of statistics, including Bayesian statistics and probability, to understand how communities assemble and what maintains diversity and ecosystem functioning, primarily on neotropical and temperate forest biomes. He's also an avid cycler, and I'm really happy that he has a little bit of time to talk us today about his work. And I hope he's going to talk us about his recent paper that's going to get published in Nature very soon. So thank you, David, and take it away.
Thanks for the introduction, Sergio. Thank you, everyone, for the opportunity to come here and speak today. I really appreciate it. So all of the data that we're going to look at today comes from an area of central Panama in the canal zone. And in that canal zone, there is an island called Barrucolorado Island. And we use data from this island because theoretically it's an old growth forest. So it's what we like to think of as, like pristine, primary, stable forest that doesn't have poaching, doesn't have a lot of human influence on it. So we can see what non-human influence forests are doing. In the middle of that island, there is a 50-hectare plot that is 1,000 by 500 meters. And within that plot, our advisor, our PhD advisor, Dr. Stefan Schnitzer, has marked and mapped, measured, identified the species, all of the liana stems in 2007 and again in 2017. And this is what the STEM map looks like. So there's a lot of them. There's almost 100,000. And arguably the largest conceptual questions in ecology relate to species diversity and distribution and coexistence. We can use these Liana data to test some of these ideas. For example, I'm going to show you a figure where we have the abundance or the number of individuals that make up a species on the y-axis, and the x-axis we have all of the Liana species in the plot ranked from most common to least common. And what you'll find is this really classic, what we call abundance distribution of species pattern, where there are few common species and many rare species. And in fact, in the Liana community, half of all of the individuals in the community come from just the 12 most common species, which means that the other half of the individuals are divided among the other 161 species. So a common question you get in ecology is, why do rare species persist? And why are there so many species in here that have very few individuals? We can rephrase that as and formalize that as what maintains diversity. And this has really been one of the longest standing questions in the natural sciences in general. You can go back to Darwin's survival of the fittest. And in the early 1900s, between Grinnell and Goss, describing the struggle of existence, they say that no two species that are similar can be put in the same space without one eventually outcompeting the other. So what this means is that these ideas kind of culminated to state that species must differ, or they must specialize to not go extinct, because if they're exactly the same as another species, one of those two species will crowd the other one out. So what that has led to is the question of, how do species coexist? And Evelyn G. Hutchinson really kind of formalized this in the 1960s and proposed this question of, how can hundreds of plankton species coexist on essentially the same light and trace minerals for nutrients? So really from this point on to today, many ecologists have been studying organisms to find slight differences among them to explain how species coexist. And species coexistence, this idea, is one of the central themes in ecology. But the problem with this is that we make the assumption that species coexist in the first place, saying all these species are here, they must have some differences because they must be coexisting. But even today, we don't actually know if they are. And recently, studies have shown that you can actually get these kinds of community composition patterns from pure neutral random processes. And what this means is that species aren't coexisting, and differences among species actually don't matter because it's all just random processes. So really, I like to step back and say, do species coexist? I think is probably the most important question, one of the most important questions that we can ask right now. But ecology just isn't just about species coexistence. I know I've been talking about this a lot, but we can also look at how communities impact their ecosystem. So we can ask lots of questions, which is why I really like this field of ecology. So we can ask, like, what is the impact of these species on their ecosystem in general? And for this talk, I've divided into three major sections based on manuscripts of investigations that I've been working on to address these three general ideas. So today my talk is going to be called Local Population, Community, and Ecosystem Processes Shaping a Tropical Forest Plant Community, which is a pretty wordy title. But basically, we're going to look at frequency-dependent assembly processes determine tropical plant species coexistence and relative abundance, which addresses do species even coexist in the first place? And then we can look at the demography of a Liana community in a neotropical forest, which addresses how these species may be coexisting. And finally, we'll look at increasing Liana density, reduced tropical canopy height, and carbon storage in an old growth neotropical forest, which addresses the impact that this community can have on their ecosystem. So first, frequency-dependent assembly processes determine tropical plant species coexistence and relative abundance. And this is a paper that I was working on from my PhD, which has been provisionally accepted in Nature Ecology Evolution. And really, the idea is we're asking this question: are species coexisting stable? Or is there just random neutral movement of species in and out of communities? And the diversity we see is really based on just random chance. The species that are there, they're always changing. Which species are there, aren't there, their abundances, how many of them there are, how few of them there are. One of the recent advances in this idea is called modern coexistence theory. And I did a lot of literature looking into how people have studied this, and I pulled out kind of 4 essential processes that have to operate for species to coexist. And what I've noticed too is that these processes operate at very local scales of just a few meters. So we have 4 criteria necessary to test for coexistence. And I want to reiterate that here you have a big patch of forest, and we can take a little section of this forest and find out within this little subsection of the community how these processes are carried out. So the first process is called positive invasibility. And this is really the idea that a species can recruit into an area, it can establish, and it can proliferate. And what this phenomenon does is it protects species from going extinct in a community. Now, opposite to that is a process we call negative frequency dependence. And really what this is that species must have some limit that caps their ability to increase in abundance. So here we can see our yellow species is really abundant in this local area. And what you'll find is that their mortality rates will increase, and they'll slowly reduce their population size over time. The third concept is a really difficult one. It's that this negative frequency dependent process has to be stronger for conspecifics or individuals within the same species than it is for heterospecifics or individuals from other species. So like we saw before, our yellow species is really abundant. Its mortality rates end up being high. But what can't happen is that it also elevates the mortality rates of other species like our green one here, because what you're left with is a local community who has the same composition of species and abundances relative to each other. It's just thinned out, and this is really just what we call like stand thinning. So our fourth criteria is equilibrium frequency. And the equilibrium frequency is the species abundance relative to every other species in the community when their population growth rate is 0, or when their population is not changing in size. So the population is not decreasing, nor is it increasing. It's kind of the stable point. And this equilibrium frequency in the context of a community needs to correspond to a species commonness within the overall community. And looking through the literature, paper after paper comes out on the maintenance of species diversity using coexistence. But what you often see is that these ideas are tested using survival or mortality. But coexistence theory is based on population change. When you really think about it, mortality really only matters in the context of recruitment. And in this investigation, I'll show you that mortality alone is not necessarily the right metric to assess how populations are changing. Okay, so I'm going to formalize this a bit more mathematically with some other figures, because I personally like the cartoons that we saw because it helps me conceptualize things. But to really understand this, it's better to use some figures. So you'll see a lot of figures. They'll more or less be the same. You'll see population growth rate on the y-axis as the response to frequency, or the relative abundance of a species within within these local communities. So here's a line that represents population growth response to a range of relative abundances or frequencies for our yellow species that we saw in my illustrations. So what we can note is, first, we have positive invasibility. So at low frequencies, population growth rate is positive. And this line is a negative slope, which shows that there's negative frequency dependence. So as the frequency of the species increases in these local areas, its population growth rate decreases. If you were to look at the effect that this species has, say on the green species, its frequency or its relative abundance would have no effect on the green species population growth rate. And fourth, you can see that there's what we call the equilibrium frequency, right? So at some point, the line crosses where there's 0 population growth rate at some frequency. So this is just one species, but when we work with communities, there's a range of species. Here, the species in brighter colors are more common in the community, and darker colors represent rarer species in the community. You can see that all these species have positive invasibility. They all have negative slopes. So they all have some level of negative frequency dependence. And if you were to plot each species frequency effect on all the other species population growth rates, you'd find that they have no effect on other species. And ultimately, these equilibrium frequencies correspond to relative abundance or commonness in the community, where the equilibrium frequency for the yellow species is much higher than the equilibrium frequency for the darker purple species. And so the reason that I have these 4 coexistence criteria is because you can essentially use a single modeling approach to test whether species are coexisting, whether there's no predictability within the community. So we can take our Liana, go back to our Liana community. We can divide this community up by quadrats and measure the relative abundance for each species within each quadrat. And then we can take that and give us a range of relative abundances that allows us to see how population growth rates respond across that gradient of relative abundances. So we were able to do this for 86 species that had enough replication for us to analyze. And here's our results. So again, we have annual instantaneous population growth rate on the y-axis as a response to the log frequency or relative abundance. So this is just population growth rate response to frequency. When we add the species in, we can see this is more or less exactly what we'd expect to see for coexistence to be happening. There's positive invasibility for all species. So when they're at low frequencies, they have high population growth rates. There's negative slopes for all species, so they all have some sort of negative frequency dependence. And we can see that they all have some sort of equilibrium frequency. And this graph can be a little bit hard to tell how these play out, but we can break these down and kind of zoom into each of our criteria specifically. So here's a set of figures where on the y-axis, we have the species in order of most common at the top and in yellow, the least common at the bottom and in darker purples. And this is the value for positive invasibility on the x-axis. So what you can see is that all species have positive invasibility. We can look at negative frequency dependence, or the negative slope of the population growth rate response to frequency. You can see that every species is less than 0 or left of the 0 line. So every species has a negative frequency dependent. And if we do a meta-analysis on top of that, we actually find that the negative frequency dependent effect is stronger for rarer species than more positive species.
Características de los servicios administrativos y financieros de los centros poblados. Asimismo,
Sorry, David. Thank you,
No
everyone,
worries.
for keeping their microphones off. Yep.
And these black distributions here represent the population growth rate response for all other species except for the conspecific or the focal species. And what you can see is that the frequency that each species has on all the other species is substantially less than what it is for individuals of the same species. And finally, we have the equilibrium frequency, and everyone has a positive equilibrium frequency. And we can see that equilibrium frequencies correspond to relative abundance or commonness in the community. So more common species have higher equilibrium frequencies than rarer species. So what this shows us is with a single model, in one figure, we can determine if species are coexisting. I like to say that, but often people have a healthy level of skepticism, including myself. So there's a lot of extra tests that we can do to really see if this is due to chance or not. So what we can do is we can take a species and say it's distributed in these 3 quadrats that I have circled here. So you can take the population growth rate response, and we can lay the frequency over the top of it, or the relative abundance within those quadrats. And what we can do is we can slowly shift those or spatially dissociate the population growth rate response to frequency in a different area. And what this will tell us is population growth rates really responding to frequency within these small localities? So here on the y-axis, we have the degrees of shifting, spatially disassociating the two variables. And the x-axis is a log likelihood of how well the models work. So further to the right is a better fitting model, a better predictive model than one to the left. When we add in the species values, we see that as soon as you shift even 5 meters, spatially dissociate where the frequency and the population growth rate responses are being derived from, that model performance tanks. It really doesn't work very well. So what this tells us is that the population growth rate response is responding to the frequency in that small locality. And this is just one species, but I can show you the same for all 86 species, have a really similar pattern. And again, what we can do is population growth rates are really recruitment and mortality rates. So what we can do is we can break down population growth rate into its constituent per capita mortality and recruitment rates, which are on the y-axis, and we can see how that responds to frequency. And so in the dot dash line, we have mortality rates, and you can see mortality rates monotonically increase with increasing frequency, but also recruitment rates drop quite sharply with increasing frequency. And at some point they cross or intersect where they're in balance, and that is where your population growth rate is 0. And we can do this for all of the species and look to see all these species have the same pattern. Now what we can do is we can take the frequency of a species and we can randomly reassign those calculated recruitment and mortality rates. So we can take our species that's in these 3 quadrats, these 3 localities, and we can take their recruitment and mortality rates and we can shuffle them, and we can see if there's some sort of legacy effect of population growth rate to frequency. So again, we have a very similar graph to before, where we have population growth rate on the y-axis and the frequency on the x-axis. And this is the observed data. And when we shuffle everything and decouple frequency from population growth rates, we see that there is no response, which tells us that this negative effect is there's actual real deterministic process happening here. This isn't something that happens from random chance. And again, when we look at all the species, you see a very similar story. And then a final test is when we get rid of mortality and look at just recruitment, what can happen is at these really low frequencies, you can have high recruitment rates that could potentially just be an artifact of chance. Because when you take this plot, and let's say we have a species that's distributed in these 5 localities, if an individual immigrates from outside the quadrant, the chances of it landing in one of the quadrants already occupied is pretty low. It has a pretty good chance of landing somewhere else. Additionally, individuals that disperse, like new recruits that disperse from adults within the plot, have a pretty good chance of landing somewhere else in the plot too. So what this can do is this can overinflate the per capita recruitment at these really low frequencies. So you can test this and say, given an adult tree or an adult individual, what is the dispersal distance? Well, we can calculate this, and it's not just random, because we know that they're, for example, like if it's a seed that's dispersed by an animal, most of the animals will congregate toward it. They'll disperse seeds, and you'll get some sort of dispersal probability where at very short dispersal distances, there's a high probability of a recruit landing. And as you get further and further away from that parent individual, there's less and less probability of a seed or a new individual recruiting. So what this can look like is if we highlight one of our quadrats, one of our localities where an adult is, it gives you kind of the spatial dispersal probability around it. And so what we can do is we can randomly assign all of the recruits to an adult and then redisperse them out based on this dispersal probability. We can do this for all the localities. And so what you get is a figure like this, where there's the posterior probability density, basically the probability of a value being true on the y-axis, to the values which we're calling the coefficient of frequency dependent effect, or the how negative that slope is for the model. And when you plot here in color is the observed data, and black is this random dispersal data, you can see that the random dispersal data does not create the same effect. In fact, dispersal distances would have to be more than double what the observed dispersal distances are for this effect to take shape. And when we look at all species, we have pretty much the same story. So what this tells us is that these areas where the frequency is really low, this per capita recruitment isn't artificially elevated due to just random dispersal. There's actually real deterministic processes happening here. So with that, our data are consistent with all 4 of our coexistence theory criteria, and they indicate that the Liana community is structured primarily by deterministic processes, which means that species coexist and this community is relatively stable. What we can also find out from this is that rare species do not go extinct because they have a strong corrective response to deviations from their equilibrium frequency. You see those slopes are much more negative, so when a rare species becomes really, really rare, its population growth rates are really, really high. And also, a really interesting outcome of this is that we find that rare species are rare due to intrinsic or internal processes rather than external processes that we conventionally think of like competition from other species, meaning that rare species are rare because of some sort of internal issues that they have, be it host-specific pathogens or something like that, versus being competitively inferior to other more common species. And so I showed you this figure of the per capita mortality and recruitment rates for all species on their own. But when you look at this from a community standpoint, we have per capita mortality on the top and per capita recruitment on the bottom as a response to frequency. When we look at recruitment, you can see this really clearly defined pattern. There's high recruitment at low frequencies, and that recruitment level decreases with increasing frequency. However, if we were to just look at per capita mortality rates at a community level, they really don't tell us much of anything. A lot of species are increasing mortality rates with increasing frequency, decreasing mortality rates with increasing frequency, or no change. So really I just want to highlight the importance of looking at the actual combined effect of these two things, which is population growth rates, to really understand how populations are changing. So now we know how they can exist, how each species can stabilize itself. Now we know that species do exist, so now we can look to see differences between species to really address how they can coexist. So in this chapter, Demography of Liana Community in Neotropical Forests, we ask these questions. Demographic diversity. So is there a range of differences in vital rates among Liana species? Right, because this range of differences in vital rates will really tell us like if there are different life history strategies between species that could allow them to coexist. So, and then is there a pattern to these differences? So what we can do is, again, on the y-axis, we have per capita recruitment or mortality. On the x-axis, we have species, the most common on the left to the least common on the right. And when we plot recruitment as positive values, and then mortality as negative values, it can be a bit difficult to see, but one thing we can take away from this is that a lot of species are doing very different things, which is a good initial sign that species have different life history traits. All the species are here coexisting because they do things slightly differently. We also want to see if there's a pattern to this. So what we could do is, on the left here, we have relative recruitment on the y-axis to their rank abundance going from most common species to least common species on the x-axis. And we can model this And we can do the same thing with mortality, and we can model that. And what we can see is that less common species have higher recruitment and mortality rates in general at a population level. So what does this tell us? Is there a range of differences in vital rates? Yes, there's a very broad range of vital rates, meaning there's a very broad range of recruitment and mortality rates that each of these populations experience. And is there a pattern to this? Yes, we see that rarer species have both higher recruitment and mortality rates. And thus, we can kind of deduce that these faster turnover rates are also what can allow rare species to respond stronger to deviations from their equilibrium frequencies, which simultaneously keeps them very rare within the community, but also keeps them from going completely extinct. And like I said, there's the nice part about ecology is you can ask a lot of questions, and these two first ones have really been about coexistence. But I want to shift the gears to something a little bit different, to looking at like how these species really, this community really affects like ecosystem level processes. And we did this in this manuscript called Increasing Liana Density, Reduced Canopy Height, and Carbon Storage in an Old Growth Neotropical Forest. So tropical forests are 40% terrestrial carbon. I don't think that there's probably not too many people out there anymore that don't realize that tropical forests are really important for the global carbon cycle. And interestingly, in tropical forests, most of the carbon is in above ground biomass. And seen here by this tree, I always have a hard time with this because it's hard to really capture the immense size of some trees in forests. But it's a good, it really helps when the tree falls down and you can see like the gap that they've left, right? So some of these trees get really massive. And height, I'm going to argue, is the metric of carbon storage. And really, it's because tree trunks get substantially taller as they get wider, right? They do this both at the same time, but they get so much taller for any given diameter. For example, the average tree with a 5-centimeter radius is 10 meters tall, making it 40 times taller than the radius square. So if we can really capture heights or changes in heights, we can get a much better estimate at how carbon dynamics are changing. And there's a few ways that you can measure height. Commonly in North America, it's depicted as like you can measure height from the ground, looking at, you know, the angle to the top of the tree to the distance that you have it. A little trigonometry will get you the height. But tropical forests are usually pretty dense. As I hope you understand now, they're also covered with lianas. So measuring from the ground is really quite difficult and labor intensive. But new technologies allow us to measure canopy height from the air. And we can do that with aerial LiDAR or using laser beams to make these topographical maps. And this is what an aerial LiDAR forest map looks like. So we can use multiple mappings through time to really assess changes in overall canopy height. And what I like to do is I like to stratify the data and look at canopy heights and all these, how canopy heights are changing in all these various areas through time to see if what areas are increasing, what areas are decreasing, what areas are staying the same. So theoretically, in a completely stable forest, on average, all height categories are neither losing nor gaining height, right? So if you look at areas with low initial canopy height on the left on the x-axis to areas of high initial canopy height, so really tall areas of forest, like how are they changing through time? Theoretically, everyone should be, all these areas should be staying more or less the same. But in tropical forests, because they're quite dynamic, what we'd expect is that canopy height should be balanced by gains in areas that are low canopy, that are increasing through time, right? The losses in high canopy. As canopy trees fall in these really high areas, they'll generally lose canopy height. But overall, this will end up balancing out as low areas get higher and higher and recruit into these higher canopy height classes. So what we can do is go back to the same forest on Barrow, Colorado Island, and we can look at the average canopy height for each quadrant, which is each of these localities, these 20 by 20 meter localities. What you can see is there's some areas that have really low canopy, some areas that have moderate canopies and some areas that have really high canopies. What we can do is look to see how those areas change in canopy height through time. What you can see is there's areas that had a lot of canopy height loss and areas that had a lot of canopy height gain. So what we can do is we can break this into a bunch of categories based on initial canopy height and look to see in each one of these areas what's happening. So what I'm showing you here is that the mean canopy height decreased in every height group. So we have the relative change in canopy height on the y-axis to average canopy height in meters across these 10 different categories. So initially very low canopy areas on the left to higher canopy areas on the right. But what we found is that the tallest areas lost the most height. So the areas that are the tallest are losing canopy height faster than areas that are shorter, even considering when you relativize for them having more canopy height to lose. So we can ask the question, why is tropical forest canopy height decreased? We know in this exact same plot that lianas increased in abundance by almost 30% across almost the same time period. So what we can do is we can look at a map of liana density change, or how lianas have increased or decreased in these same quadrats, these same localities. And we can model the effect of the liana density change on the relative change in canopy height. What we can do is we can use a special type of model, a special type of hierarchical model called a partially pooled model, which will give us not only how has it changed across the whole plot, but how has it changed in these different initial canopy height groups. So we can see if lianas are changing canopy height at the largest canopy height sizes or in very low canopy height areas. So what we found out is that as liana density increased, canopy height decreased overall in the plot. So on the y-axis, we have the relative canopy height change as a response to relative liana density change. On the x-axis, it's pretty significant. It's almost, the credibility is almost 100%, which is about as good as it can get. And we look at each of our 10 canopy height categories going from the lowest initial canopy height in the upper left across, and then the highest canopy height areas in the bottom right. You can see that in every area, so across the entire forest, regardless if it was an initially low canopy area or initially very high canopy area, lianas, increasing liana density caused a decrease in canopy height. And what this means is we can look at total carbon forest change and show that as liana density increased, forest carbon decreased. So what we see here is on the y-axis, we have above ground carbon change in log megagrams on the liana density change. And when we look at each of our 10 initial canopy height groups, lianas have this effect everywhere in the forest, in all of these canopy height groups. So what is this telling us? So increasing liana density, reduced canopy height, and carbon storage in an old growth neotropical forest. So in this forest, canopy height is decreasing. Increasing liana density is decreased this canopy height, or at least is strongly correlated with it. And lianas are increasing in tropical forests, mostly in the neotropics, which may exacerbate canopy height and carbon storage losses. So currently, tropical forests are a carbon sink, absorbing more atmospheric carbon than they emit. However, decreasing canopy height could convert them to a carbon source, meaning that they'd be emitting more carbon than they absorb. So this is a lot of information. So my overall summary, what I want you to take away from this is that the liana community is stably coexisting through local deterministic niche-based processes, and that species coexistence must be tested using population demographies. Liana species differ in their demographics, suggesting that liana community has a range of life history strategies allowing them to coexist. And lianas are increasing in tropical forests, driving canopy height losses, reducing tropical forests' ability to store carbon, And with that, I'd like to thank Sergio for inviting me here, everyone for your attention, my lab, my collaborators, my funding. You can reach me at my email. And with that, I'll take any questions.
Wow, David, okay. That was a lot, and that was really quick. That was pretty intense. It's like grabbing a 9-volt battery and just putting it in your thumb and like believing that the force is not going to collapse.
Awesome.
All right. Okay. So I have my one question, but I have other questions.
Sure.
More on the yeah. Okay. Let me start with the first question. Okay.
Awesome.
I know your parents. How do you
Mhm.
explain this to your parents? How do you explain this to someone that knows the forest, okay, that probably has walked through a tropical forest, maybe not. I don't know if it really matters. How do you explain something so complicated as coexistence and all those four
Criteria.
criteria of coexistence to someone in a second semester botany class?
That's a great question. So generally, I like to break them down. Yeah, it was a little fast. Maybe, sorry. It was, I could have slowed down a bit.
No problem.
Generally, I like to break those down. It's really like your first criteria, which was positive invasibility, is really like the weediness of a
Okay.
species. Like how weedy is it? Or like think of, you find a mouse in your house. You find one and you leave it alone for a couple of days and suddenly there's 10. And you leave it for a week and there's
Okay.
100, right?
All
Really
right.
high positive invasibility. When
Okay.
there aren't any, suddenly there's one, then there's a whole bunch, right?
Okay.
And the really the negative frequency dependence, like the cap, like is this idea that if one species could just keep expanding like mice in your house, eventually there would be nothing left but mice in your house.
By mice.
So at some point, there has to be something. If a community is coexisting, there has to be something that limits all species maximum population numbers, right? And
Okay.
really, I like to put this to, if you look at any farm or any agricultural system,
Okay.
farmers spend a vast majority of their time making sure that monocultures don't die. And they
Okay.
can put as many of the exact same plant in as small amount of area as possible. And that's essentially agriculture. And we force that to happen because naturally it won't happen on its own. Some plague, some blight, some insect will come through and decimate the population because it's at such a high unnatural density.
Okay. And so that negative frequency dependence is the more common you are, the more prone you are to die.
Yeah, I mean, like the one that lots of people like to use that I guess I'm going to use here, I generally don't like using it, is if you think about the pandemic, is most issues, most cases happen in the most densely populated areas, because as density increases, population sizes increase in an area, specific diseases transmit much more easily, is one way to think of it.
True. All right. That's
You can think of it,
second.
herbivores, any of that kind of stuff.
Okay. The third one.
The third one is it's really the difference between this effect controlling a population or what we call like stand thinning, which from like succession work. So if a species is around it and it reduces the number of individuals from its own species, but also reduces the number of individuals from other species, you end up with like the relative abundances. So the relative numbers don't change. It's just the absolute numbers, it's go down. So instead of 10 individuals and 6 individuals from species A and B, you have 5 individuals and 3 individuals. So it's just a thinner forest. And that's usually what you see like in success. It's a good indicator of successional processes, I like to think of. Just you have lots of individuals, everything grows up, forest gets thicker, gets a nice canopy, and then individuals start thinning out. You lose density per hectare over time.
Yeah.
And then the equilibrium frequency is really like the culmination of all these, and what really indicates whether a population can stabilize, right? Because in localities where it has really high density, it has a negative population, meaning through time its population is shrinking. But in other localities where it has really low population size, there's a positive population growth rate, meaning that its numbers are increasing through time. And on average, overall, over a like a plot or a landscape or a region, that net effect, that average effect, ends up balancing the species out.
Okay. And so given what you just said, I'm really puzzled by the second part of your talk. Then why species are not going extinct if the rate by which mortality is so great and recruitment is not in so many species? Why they're just being rescued by whatever is going on outside? Or am I completely misunderstood?
Hey, I'll take you back
Please do.
to this
There.
figure.
Mm-hmm.
So this is all of the 86 species, and it's hard to tell which line is solid and which one is dotted, but generally like the downward sloping line is the recruitment rates, and the more horizontal or slightly positive slope line is the mortality rate. So you see at low no is mortality rates. So if we go back, I'll zoom back into just one species.
That one.
So you see at low frequencies, the per capita recruitment rate is much higher than the per capita mortality rate, like 0.8. So there's a lot of pressure. There's a lot of possibility of driving the species at low frequencies to increase its frequency.
Okay.
And then at some point when its frequency is really high, the recruitment rate dips below the mortality rate. So mortality starts outpacing recruitment, which results in a negative population growth rate or population contraction. So you start losing more individuals than you're gaining.
Right. It has to be further right. If he's closer to the left, then it's going to be a problem. Okay.
So for what this really means is in any locality where the species is not very abundant, like its ability to increase in abundance is really strong, and vice versa in areas where it's really common.
Yeah. But then
so keeps wanting to go to this middle point.
Okay. Right. Okay. I have more questions, but Juan just turned on his camera, meaning that he wants to ask a question. Juan.
All right.
Yeah, thank you. Actually, I couldn't find where to raise my hand on the new Zoom menu, so I see a little heart, and it's kind of weird. But thank you, David. That was really interesting.
Thanks.
I'm still trying to work out my question, but it's kind of an obvious question that I'm sure you have thought of, given that I'm a big fan of functional ecology, trait-based ecology, and
awesome.
So it seems you have the perfect setup to actually test which traits could actually influence some of the responses you're seeing. Like I think like having a very high per capita recruitment rate could be related to more species with more an ecological strategy, which is very acquisitive with very high, good dispersal, high production of seeds, and fast growth, but also die easily in the understory, versus much more conservative strategy. So have you guys worked on that, or are you working on that?
Is actually a colleague of Sergio and I is a functional ecologist, and he has a lot of functional trait values for these same Liana species and many other ones. And we're all kind of working on linking what functional traits really correspond to these different life history strategies, which is, you know, the second manuscript that I talked about in here. I was really quick because I think it's pretty straightforward, but there's a lot of different life history strategies among lianas, which for a lot of people, they find it really surprising because the old adage is that they're just these like highlight dependent, gap dependent, pioneerish. They should all just kind of line up in this like really fast acquisitive kind of traits. But we're finding, at least from like a demographic standpoint, that there's plenty that are like that resemble more like shade tolerant tree species.
Great.
But it's there. our fingers are
That-
crossed that
Yeah,
lines
that's
up.
great. Yeah. Well, because just maybe it can be of a help. But with one of my PhD students, we look at functional trait from anatomy, wood anatomy, and relate to
Yeah.
mortality rates in Parakou,
Okay.
in French Guiana. And what basically we found is that the mortality rate depends on the stage. Is different from sapling than for adult and
Yeah.
seedlings. So the different traits are more correlated to mortality at
Yeah,
different stages.
which-
So it could be fun to look into it too.
Which is, yeah, really interesting too, because just like for a good number of lianas, not all, right? But like a lot of them, the leonescent, like when they become a leonescent, like a climber, is really ontological. Like they'll grow they'll look like tree saplings till they're 2.5 meters tall.
Huh.
Right. So like that ontology of like when that like we're gonna start climbing stuff flips is really
Change.
is something that's interesting, right?
Very cool. Yeah.
No,
Great.
thanks for the questions.
Yeah, thank you. Really interesting. I really appreciate it.
Thanks.
And anyone else questions? If you want to do it in Spanish, we can translate. You want to write it down in the chat.
No
Ahí-
tenemos, profe.
Okay, then I have another question, David.
Ujú.
Yes.
You don't elaborate on what are those deterministic processes. You just say we compared against our concept of random or null, but you don't point
Yes.
out exactly which are the deterministic processes that are driving these patterns, at least in the first chapter. Can you please elaborate on what
It's a good question, and it's a very specific reason I don't elaborate on it. Is because I think a lot of people, especially this is an observational study, so there's no experimental design to really pinpoint what is driving what. Like what is driving this process? It's just a matter of do species coexist? That's really the main question, right? And a lot of people will say, oh, well, it's soil pathogens, fungi in the soil that are specific to each one. That's what's happening. But it could also be specific herbivores. It could be any sort of biotic agent, which is what I say in the manuscript very specifically, because it could be a soil pathogen for one species and herbivore that lives in the canopy for another species, right? The herbivore could be an insect, it could be a mammal, it could be, maybe it's a bird species that really likes the fruits, but are like a pigeon where they have a crop and they destroy all the seeds that really limit it.
Right.
So it could be a number of things, and each one of those things could be different depending on the species. So that's why in this specifically, we're not using like any sort of like fungicides or herbicides or anything to really like tease apart what the biotic agent is, just that some biotic agent that is specific to that species is keeping it in check.
Okay.
But yeah, it could be a lot of things, right? There's a lot of stuff that happens in a tropical forest.
Okay. And so you were telling me a few weeks back, just for our audience and Juan, who's very attentive, it's really interesting that people feel that mortality is the most important variable to measure coexistence. But so I get you get a lot of pushback because mortality is being heavily used, and now you're using just kind of species population growth rates, which is kind of classic 1940s ecology. And so why not keep using something that I teach to my students, and instead of use mortality? Like, when did that whole thing kind of change, and why? Because it's just really hard to get, or what?
I think it's hard to change because it's hard to get population growth rates, especially people study the forest for trees. So that, trees are usually well into the hundreds of years old. So getting population growth rates can be really difficult. So I think a lot of people like to look at and use seedlings and then look at seedling mortality rates or survival rates, as they like to say, or growth rates, right, as proxies, because I think it's easier. But it's really interesting how, 100 years ago, it's all population. When you look at the theories, like population change is based on population growth rates. It's not based on the mortality rates or anything like that. Because I always find I just always find it really confusing. It's like, oh, this has really high mortality rates next to an adult. And it's like, yeah, but it also probably has pretty high recruitment next to that adult too, but we're not looking at that. Okay. And then, yeah, and like, especially like when they do it at like certain life stages, you know, how many hundreds of thousands of seedlings of a species die before they become a reproductive adult? Like, I think it's really, for me, like, I have a really hard time believing like increased mortality rates of seedlings in certain areas versus others is indicative of some sort of like population level dynamics, because it's like, yeah, but you know, that seedling that you measured that's 20 meters away that hasn't died yet, it's got 100 years to go before it could become a reproductive adult, like hitting the canopy and becoming a reproductive adult, or 20 years. I mean, it's really variable, right? So that's where, like, so this data set is, I just used all individuals greater than 1 centimeter in diameter. Didn't matter if they're big, small, everyone in the population, just look at what the entire population is doing. Because it's also, again, probably like what Juan was suggesting too, is like ontologically different biotic agents may be affecting them in different ways. You may start out with fungal soil pathogens being very important at small sizes, and host-specific herbivores being really important once you're an adult.
Right. Or some
but it's the
fungus
net
that's
effect.
gonna give it heart rot. Yeah. But it's the net
Right.
effect.
But it's the net effect of all of it. It could be both, right? You could have all of these things, which is probably more likely than one or the other, than some sort of mutually exclusive thing. But it's really the net, like the net effect. Because at the end of the day, that's what you want to know, is what the actual population is doing.
Exactly. All right. I don't see anyone else raising their hands. It's
No,
almost
ni en
12
el chat,
o'clock.
profe.
Perfecto. All right. Well, David, I'm really looking forward to the Nature paper. I really want to read it because I know
thanks.
got a lot of pushback, and I'm really, really happy that it finally came out. Thank you everyone for joining us on this very interesting talk about community ecology of tropical forests. And well, have a great rest of the week. David, thank you again. Say hi to Laura and Lucas for me, and your parents as well.
I will.
And
we'll look forward to seeing you.
Yeah, absolutely. Well, thank you everyone for joining us. And in 2 weeks' time, we have another talk. So please stay tuned to our emails. Thanks again. Have a good one.
Thank you.
Juan.
Chao.
Juan.
Chao. Gracias y todos.
Chao.
All right, David.
Muchas gracias por su asistencia y participación. Nos encontraremos próximamente en el seminario que tenemos programada para el 12 de septiembre.
12 de septiembre.
Bueno, muchísimas gracias y muy buena tarde, buen almuerzo. David, un gusto en conocerle. Muchísimas gracias. Buen día.
Igualmente.
Bye,
Gracias
David.
a todos.
Thank you.
Chao.
Thank you so
Hablamos.
much.
Chao.